So yes, so welcome back everybody. We're now going to start our third session, which is vaccine development, chaired by Dr. Graham Taylor from the University of Birmingham. So Graham, I'll hand straight over to you if that's okay, and we'll start the third session on vaccine development.
Okay, Lucy, thanks very much. So if you think back a year ago, just where we were, things seemed quite desperate. We didn't have a treatment or a vaccine for a new virus that was spreading across the globe. Now where are we? We've got drugs that are proven in randomized clinical trials to increase survival for the sickest patients on ITU wards, and vaccines that have been proven under real-world conditions to confer protection. But it's clear we need to do more. There are patients that won't be able to be vaccinated for various reasons, that need protection from severe disease, so we need better drugs. And of course, we need better vaccines. We need vaccines that can cope with variants, for example. So it's a real pleasure to chair this session, and I welcome our first speaker, Professor Imre Berger from the University of Bristol, who's going to tell us about a druggable pocket and a drug in the SARS-CoV-2 spike protein.
Thank you so much. I will start my talk in a moment, so let's see whether I can activate it. Okay. All right. Do you see my slides?
Yes, we can see your slides. Thank you.
Excellent. Okay. So it's a great pleasure to be at this meeting, and I heard like so many exciting things already. So I have to say immediately, I'm not an immunologist or like really a virologist. So became like many others into this arena of SARS-CoV-2 research due to the circumstances. So we are doing some vaccine work, but this is not what I will tell you about today, even though that's also very exciting. I think it's like about a thermostable vaccine based on a scaffold which we developed just months before the SARS-2 pandemic hit us. But what I will focus on today is something else, namely a fortuitous and serendipitous discovery of a druggable pocket and actually a drug in SARS-CoV-2, even though it took us a while until we realized what we really had there in our hands. And basically, I shouldn't be the person who is giving this talk, but it should be Christiane Schafitzel, who did the cryo-EM work I will show you about this, and who has received quite some recognition lately due to the research which I will present, including this cover page thing here on the Bristol magazine in this iconic picture with a self-made mask, because at the beginning of the first lockdown, which is almost too long time ago already to remember, we didn't even have masks at our disposal, so we made them ourselves from old t-shirts, and here is one of those. You can see that. Okay. So on the left, you see this SARS-CoV-2 virus, which we have seen now way too many times, so everybody is entirely familiar with. I don't need to talk about that. And on the right, you see a cryo-EM structure which we developed which we determined at high resolution. And the reason why we were doing this is that this is how we came into this SARS-CoV-2 research. That at the beginning of the first lockdown, people here in Bristol, like everywhere else, as we have heard before, were starting to do serology testing and all kinds of other experiments, and they needed antigens. And my laboratory is particularly has a platform which is particularly useful for making all kinds of complex proteins, including antigens. So we made the SARS-CoV-2 S protein and many other things also. And because we use a slightly different technique than most people use, so we have heard before from Crispin and other people that many people express the SARS-CoV-2 protein in CHO cells, so we make it differently. So we had to then test whether the spike protein looked authentic, and this we did by cryo-electron microscopy. So the two guys who were doing this work was Fred and Julien, who produced many antigens for the colleagues here in Bristol using our platform. Okay. So when we had this spike protein and we looked at it, and you can see it again on the left, so that's a cryo-EM structure at 2.8 resolution, so not too bad. It's a trimer, as you know, and you can see like the individual chains here in different colors. And when we determine the structure and we trace the protein through the cryo-EM density, there, which was like reasonably good, we found a tube-shaped density there, which we couldn't assign, which was not protein, and which has also not been seen in the other SARS-CoV-2 spikes, which had just been determined shortly before that, which did not have this density, additional tube-shaped density in there. So we knew that there was something in there and it was not protein, but we didn't quite know what it was. And this tube-shaped density was not just anywhere in the SARS-CoV-2 spike, but it was in a very interesting and important part, namely the receptor binding domain, which, as you know and as you have heard, is the one which mediates attachment to the ACE2 receptor, and which then leads to infection of the cells. And what you do when you have a cryo-EM structure of a trimer, you first refine it unsymmetrized. So we did that, and then we saw that in each of the 3 RBDs, we had this additional tube-shaped density. And then when you convince yourself that they are sufficiently close to each other structurally, those monomers, then you apply symmetrization and you get a somewhat better resolution. And you can see at the end here, on the bottom right, you can see this tube-shaped density in the RBD. And this work was done by Satish, Ufuk, and Christiane, whom you see here without that red mask. So we had a look at the non-symmetrized structure, and we found that in all 3 RBDs there, so the pink, the blue, and the green one, we had this tube-shaped density. And you also see on the left in the bottom right that the density in the pocket was very good. So you could really trace every amino acid there, and in addition to this tube-shaped density. And we were lucky enough that in my lab I have a postdoc, her name is Christine. So this work was done by Christine and Kapil in the lab, and Christine had worked with fatty acid binding proteins before. So she had seen, she's a crystallographer, she had seen these tube-shaped structures, and she had this hunch that it was a fatty acid. It could be a fatty acid, what she had in there. And what she then did is she went into the RBD, which is this data bank of like thousands and many thousands of protein structures, and she looked at various fatty acid binding proteins to look for similar tube-shaped densities there, and which looked similar to ours. And she found them 2 other proteins and several others too, which had a fatty acid in there. And there people knew what it was. They knew that it was linoleic acid. So based on shape considerations and on similarity to other proteins in the RBD, we could propose that the tube-shaped density we saw was indeed a fatty acid, and not just any fatty acid, but linoleic acid. Here on this slide, you will see again the spike protein in the top presentation, this time the 3 linoleic acid molecules assigned in the structure. But of course, this was only a proposal, so we were not sure whether it was linoleic acid. The resolution was not sufficient to unambiguously decide this. So what we did there is we collaborated with 2 colleagues in the Max Planck Bristol Center, who actually work in Germany at the Max Planck Institute in Heidelberg, Oskar and Joachim. And they are specialists in lipids, and they have all the equipment you need to analyze what you have in a sample. And they put our spike protein preparation through the analysis and could then confirm that indeed it was linoleic acid, what we saw in there. And this was confirmed to the 4th digit after the decimal, that what we had in the spike protein there was indeed linoleic acid. So now we could confidently assign this tube-shaped density, and you see the pocket on the right. And what's special about this pocket, among other things, is that, so you have, basically it's bipartite. You have a greasy tube in which the hydrocarbon tail of the fatty acid fits, and you have on the other end, you have the polar head group sticking out of this tube and interacting with positively charged amino acid residues, which form a so-called polar lid. Okay, and what was special about the spike protein was that normally in linoleic acid binding proteins or fatty acid binding proteins, both the lid and the greasy tube which housed the fatty acid would come from the same protein, but here it was coming from 2 proteins, and that will become important in a moment. So what you had there is you had the blue RBD in this picture, which housed the greasy tube in which the hydrocarbon tail was sitting. And you had a green adjacent RBD, which provided the polar lid, which was locking down on the head group of this fatty acid. And at the bottom, you see the same pocket in a surface charge representation. That's the kind of pockets people like who develop drugs, because it's a very well shaped pocket in which the linoleic acid fits very smugly. And you might want to develop something which binds even better than linoleic acid and could then interact and displace the fatty acid and interact even better with the spike protein. But now we knew that we had a free fatty acid, and that was linoleic acid bound to the spike protein. And this was surprising and had not been observed before. Okay, so why is this significant? So cryo-EM is a technique where you not only get the structure of your protein of your sample, but you can actually also look at what you had in solution. So you get like an idea of how the individual molecules, which you look at as a single molecule really in the electron microscope, how they look like. So what kind of a population you have. And what we noticed was that in the spike proteins, which have been determined previously, you had a preponderance of the so-called open form, which we see on the left. So most of the spike proteins in the previous structures were in this open form, and there you expose the receptor binding motif that's colored here in red, which binds to the ACE2 receptor. And that's a de-infectious, you can consider this as an infectious form. And we had like a subpopulation of this open form too, but most of our spike proteins were actually in this closed or even locked form, which you see on the right, because what was actually happening was that the linoleic acid would tucker these RBDs together and thereby close the spike protein and lock it in a condensed form, and hiding essentially the receptor binding motif. So it would not stick out anymore like in an open form, but it would be locked away. It's a closed form, and in this form the receptor cannot bind. So that's considered a non-infectious form, and that was the preponderant form in our sample preparation. So we then thought, okay, so if it's maybe like preventing the virus from being infectious, then it should have a additional effect when you use it together with, for example, remdesivir. So in a cell culture experiment, remdesivir like slows down the replication of the virus. And indeed, this was what we saw. So so remdesivir was a big hope at the beginning. It didn't quite work out as one hoped, but at high concentrations, you would get a replicative stopping effect or like diminishing effect, but with heavy side effects. But if you now added linoleic acid to this experiment, then you could drive down the concentration of remdesivir you needed to deploy to stop the virus from replicating. Okay, so we had this situation that we have this linoleic acid. The linoleic acid is stabilizing a non-infectious or like locked condensed form of the spike protein. It synergizes with remdesivir to stop viral replication, and actually does it also on its own without remdesivir, as we could confirm. So what is special? What is so special about this linoleic acid? We didn't quite know this, but we have a collaborator, Dan Fitzgerald, who's a specialist in free fatty acids, and he enlightened us that linoleic acid is essentially free fatty acid, so cannot be synthesized in our body, and you have to take it up through the diet. It's a key precursor in metabolic pathways, namely the metabolic pathway over arachidonic acid to eicosanoids, including prostaglandin, and those are central to inflammation and immune modulation, which become dysregulated in COVID-19 when you have the disease. LA is also essential for maintaining membrane fluidity. It's like an unsaturated fatty acid, and when it's in a phospholipid form in the membrane, then it makes the membrane smooth and fluid, and that's important for the surface tension in the lungs. And we know from other diseases that when you alter the LA pathway, you can get acute respiratory distress. So if you deplete the membranes of LA, you get respiratory distress and severe pneumonia, which are also key elements of COVID-19 pathology. Moreover, and this was Dan's specialty actually, is LA is a ligand for FFAR1, GPR40. So this is a free fatty acid receptor, very important in lung epithelial cells. It's a key regulator in insulin release, but also in lung epithelial cells, it is key to controlling inflammation. So we have both this inflammation control aspect with FFR1 and also the regulation of insulin release in the beta islet cells. And as we know, diabetes is a risk factor for COVID-19 patients. So there's again a connection. And what just before we published our paper, a very important and interesting study came out in Cell where people looked at what is actually in the sera of very sick patients, and it became they reported that the LA levels are continuously decreased in people who suffer from COVID-19. So basically, when this picture emerges that we have LA, it is like the virus is scavenging this LA, it is somehow impacting on the LA metabolic pathway. And remember the icosanoid storms, which are a key element of COVID-19 pathogenicity. And we have this connection with the diabetes comorbidity, which is a risk factor for COVID-19 patients. So when we tried to digest this, then we realized that our discovery provided the first direct structural link between linoleic acid COVID-19 pathology and the SARS-CoV-2 virus itself. And it also suggested that the LA binding pocket, this very well defined pocket in the S protein, but also the LA signaling axis and the LA pathway to arachidonic acid and eicosanoids could be excellent therapeutic intervention points against SARS-CoV-2 infections, in particular in people which have comorbidities. Okay, so how can this knowledge be used against the virus? So when we were thinking about this, we came across a precedent by Michael Rosman, who is a titan or was a titan in structural virology, and he researched rhinovirus. So rhinovirus is a much less harmful disease. It's dangerous for people who have asthma or have like some immune suppression, but what they did, like what Rosman did, is that they developed drugs against rhinovirus, and they were exploiting a fatty acid binding pocket, which was in that area, in those proteins of rhinovirus, which bind again to the receptors, are very similar to what we have in SARS-CoV-2. And what they didn't know exactly which fatty acid it is, because it was not a very high affinity binding, but what they could show is that when they developed MIIN compounds, chemical compounds, they could replace the fatty acid, distort these proteins, and then not have receptor binding anymore, and abolishing the infectivity. And this was successful in the clinic in phase 1 to 3. And so now the exciting thing about this is that they had to work very hard and do a lot of medchem to get a picomolar EC50 ligand, these MIIN compounds. In our case, however, the linoleic acid already is a picomolar EC50 ligand. So like we have, or this we just realized after thinking about this for quite a while, that we actually do have already a drug, and that drug is available in basically unlimited amounts, because it can be made out of sunflower oil, and there's 60 billion tons of sunflower oil per year being produced. So it's a widely available drug, and it could be that global remedy for this problem of SARS-CoV-2 in terms of abolishing the infectivity and the transmissibility of the virus. And these are the people behind this. I can be super happy to have them. Many of them volunteered to work during the lockdowns, and then before and after, and also until now, especially in the first lockdown, which was very hard on everybody. And I also want to thank the funders who's basically, the grants which we raided to support this research, because when we started here in Bristol working on SARS-CoV-2, you could only work on SARS-CoV-2, so we had to, take this funding and then channel it into this pivot, which we made like so many others. Thank you for your attention, and I'm happy to take questions.
Thank you, Professor Berger. That's absolutely fascinating. I've just got time for one very quick question, which would be, do the other coronaviruses also bind fatty acids in their spike, like SARS-CoV-2? Okay, so the situation is like this, that nobody knew this before. So like all the structures that you find in the PDB were analyzed before had no fatty acid binding in there. But we, of course, now looked carefully at that, and already our results, which we published last year,
showed that the fatty acid binding pocket is conserved in SARS-1 and also in MERS, and actually in every one of the mutants which are coming out. This is something quite interesting, that of the thousands of mutants which are occurring all over the globe, including like B117 and the South African one and the Brazilian one and the others, none of these mutations actually like hit the pocket. So the pocket is there in all of the variants and in MERS and in SARS-1. And SARS-1 was in 2002. So, it has been conserved, and we have now also experimental evidence that they bind not every fatty acid, but actually linoleic acid.
Thank you. Absolutely fascinating. In the interest of time, we'll move on to our next speaker, who's Professor Lindy Durrant from the University of Nottingham. And Lindy's going to tell us about a new RBD and nuclear capsid COVID-19 vaccine that targets dendritic cells in vivo, stimulating potent T cells and neutralizing antibodies.
Hi, can you all see my slides?
Perfect, thank you.
Yeah, no problem. So, hi, I'm Lindy Durrant. I'm Professor of Cancer Immunotherapy at the University of Nottingham. And as you say, I've moved from cancer to COVID for this short period of time. But I'm also the Chief Scientific Officer of Scancell, who are co-developing this vaccine that we call Covidity. So all the team here at Scancell have helped design and screen the vaccine, but it's actually a UKRI grant, and it's in collaboration with the University of Nottingham. So James Dixon is providing a new peptide delivery system for us, and Jonathan, Janet, Chris and Richard are virologists who are giving their expertise and also doing some of the screens. We also collaborate with Graham at Nottingham Trent University. He provides the animal models for us. We deliver the vaccine either with electroporation with ICOR or needle-free injections with PharmaJet. Our viral challenge model is via NIBSC, and basically our development people who make the vaccine are COBRA. So this is a DNA vaccine, and it's a DNA vaccine incorporating two proteins. And what we've chosen to do here is just take the RBD of the spike. As you all know, that's the region that actually binds to the ACE receptor. But we've also incorporated a second protein, which is slightly different from most groups, and that's the nuclear protein. And our idea here was to not only induce viral neutralizing antibodies, but also T cell responses against both the S and the N protein. And the idea there a little bit was to try and prevent escape variants, because we have two antigens that have to mutate, to give us durable memory responses, because we'd actually used a very similar vaccine approach in a melanoma study. And in the melanoma vaccines, again, we have two different antigens clearly from melanoma rather than from a vaccine. And we had 6-year stability study on this vaccine. It's very stable, it lasts forever, and we get great safety responses. And in the melanoma, we mainly stimulate T cell responses. So the whole process of this vaccine was to stimulate very potent T cell responses. Again, very briefly, this was the melanoma study. So each of these red dots here is people who are getting recurring melanoma. So it's removed by surgery, it comes back, it comes back, it comes back. Very distressing for these patients. At the actual time of this study, the surgeon thought they'd removed all the lesion, but clearly there's a very high risk of recurrence, as you can see. If you look at the green bars, these are all people that received this DNA vaccine. They had no further recurrence whatsoever, and they're disease-free, in fact, potentially cured of their disease out at 5 years now. There was 4 patients that did get some recurrence, but then responded beautifully to therapies, particularly the checkpoints, and they're still alive. Unfortunately, there were 2 patients, despite the vaccine and despite other treatments, actually succumbed to their disease. So at the end of this study, we had 14 out of 16 patients alive. And what you would have expected here, between half and 2/3 of them to be dead, i.e. like these 2 patient cohorts, these 2 patients. So the big study was no toxicity despite numerous injections here, and we got very strong memory T cell responses. So the idea was, okay, can we use the same vaccine, the same DNA, and incorporate, I'd say, the spike RBD? We would get the neutralizing antibodies to prevent the virus getting into cells, and the nuclear protein mainly to stimulate good T cell responses in case any of the virus had actually got in the cell, that we could actually get rid of it. We have enhanced our vaccine now to give even better responses, and I'll tell you a little bit about that. And as I say, we are also developing a non-toxic peptide delivery system, but I won't talk too much about that today. Very quickly, mechanism of action. The DNA can either go directly into a muscle and the muscle will produce the protein. So either as an FC fusion, which will then get internalized via the FC receptor, gets degraded, the T cell epitopes get released and they get presented on MHC1 and 2 and we stimulate T cell responses. But interestingly, the advantage of the DNA over just using some sort of protein vaccine is it can also go directly into an APC. It can then synthesize these protein and go through the two mechanisms. And in a wide range of studies in the melanoma vaccine, we showed that this cross-presentation and this direct presentation gave us a 2-log improvement in avidity of the T cell responses. So it's a really good thing to have both. Clearly for this vaccine, we also needed B cell responses or antibody responses. So some of this protein needed to be functional, intact, good, well-folded protein. So we would get antibody neutralizing responses. So they're either as the whole protein or as an FC fusion. Again, we tried multiple different ways of presenting this, either as a monomer, FC, as a trimer, in various ways. And we had 2 different size RBDs, either the full RBD or a slightly shorter version. And again, with the nuclear capsid, we either had the monomer, we had FC, or we had this modified FC. And we had, as you can see, a whole range of constructs combining each of those things. The assays are fairly straightforward. People have gone through them already. It's a straight ELISA against the whole S1. So although it's an RBD vaccine, we screen against the whole S1 or N protein. We have neutralization assay, either a surrogate neutralization, just blocking binding to ACE2, or again, Richard's done a beautiful job with the pseudovirus neutralization assay. T cells we do as either ELISpots with or without blocking or intracellular cytokines. This is just showing you some of the neutralization assays. So this is all take the vaccine, immunize mice, take the sera and have a look. Hopefully what you can see here is the open circles, or if you had a vaccine with just the whole spike protein. So these are the current approved vaccines. And what you see is the short RBD is not as good, or it's very similar to the spike, but if we use the longer RBD, we're getting nicer immune responses, and that's just represented here. Either the monomer or the FC gave nice high antibody responses here. Clearly the RBD monomer is simpler, so that's what we've gone for. Again, we do get antibodies to the N protein. Again, probably the best one is the monomer. There's not a great deal of difference between here, and as I'll go on to show you, this FC variant here gave better T cell responses, so we've tended to go with that one for the N protein. Again, just showing you some neutralization data from Richard. You can see the trimer wasn't great, the RBD-FC wasn't that great at neutralization, the modified RBD was better, but by far the best really was just the simple monomer. It's giving very high titer neutralization assays here, presumably because it's folding the best, it gives the best antibodies, and that's what we've gone forward with. And again, we've done a direct comparison here with the vaccine that contains the RBD versus the whole spike protein. And again, you can see we get significantly better responses with the monomer, giving us a much better ID50 here. In terms of modifying that FC, what we'd already known is that mouse Ig3 antibodies are very good when you bind them to high density antigen. They have these what we call no covalent association, and they form these clusters. That makes them very stable on the cell surface, and it can improve internalization. Unfortunately, when you make the mouse antibodies into human antibodies, or you do it on a G1 background, you lose this activity. So what we actually did is we mapped the sites within mouse IgG3 that were causing this effect, and we transferred them to human G1. And what we found is there's 23 regions, 23 amino acids here within this elbow region of an antibody, that if you confer those, if you put them from the mouse to the human, you significantly improve the affinity of the antibody and you get this clustering. So in essence, it's the same sort of idea now, but if other than just having monovalent targeting of CD64, we get this beautiful homophilic interaction now that significantly improves internalization, and we hope that then leads on to better T cell responses. Again, if you want to just do some biocore data here, this is just a normal FC, basically showing you on and off rate. Hopefully you can see the modified here is we get not only much higher binding, but particularly high density. This off rate is extremely slow. these responses are very high affinity. Does that respond? So here we're looking at responses to N. This is a particular N epitope in HLA-A2 transgenic mice, so it's an A2 epitope. You can see the nuclear protein works. If you make it an FC fusion, it's slightly better, but by far the best is this modified version. Basically, you're getting significantly better than FC and the monomer, and you're going from about 150 right up to 500 spots, a fourfold increase. If you look at an N-peptide pool, so you're looking at a whole range of epitopes here, it's even more dramatic. And although the N and the NFC do give responses, the modified gives even better. Interestingly, if you put the RB monomer alongside this NPFC variant, you also get a really strong T-cell response to the RBD monomer. And that's because internalization via CD64 actually activates your dendritic cells. So you're also getting better presentation of any linked epitopes. And again, hopefully you can see that's better than a whole S spike protein, giving us some hope that maybe this vaccine would give us as good, if not better, responses than the current vaccine. In terms of other epitopes, we've done similar to this. As I say, we've screened HLA-A2 transgenic mice with the peptide pools, RBD or N. We've identified individual epitopes. We've shown some of these epitopes are blocked completely by CD8, so we're getting CD8 responses to both the N protein and here blocking with CD8 to an RBD, so CD8's in both. Again, if you look in biopsy mice, again, you see nice responses to the pools. This is a different epitope, clearly because it's a different HLA molecule, and this is predominantly a CD4 epitope. Again, if you look in the C57s, we've done all these in all of them. Basically what you're seeing is we see nice gamma responses to N peptide pool, N epitopes or RBD epitopes, a little bit of TNF alpha, but not huge, and no IL-4 and no IL-10. Okay. Very similar story with CD8s, again, predominantly gamma with a bit of TNF, but no IL-4 or IL-10. So this is the vaccine that we're taking forward now. So it's the RBD monomer linked in the same vector to the N protein with this FC modification. It's called SCOV1 or scancell-CoV1 or SN15. I do that because many of the group working with me are used to calling it SN15. As I say, we've got great safety and toxicity data, so the MRHA is saying we don't have to do new toxicity, we can go straight into a clinical trial, which is great. The manufacturing is underway at Cobra and should be finished shortly. We then have to do the release, and we're hoping to start this vaccine first half or early beginning of the second half of this year. The trial will be, the first cohort will get the vaccine with electroporation. It's electroporation we used in the melanoma trial, and it's extremely good at getting the DNA uptake. It's not that useful for a pandemic because you have to have a device in every center, so that would be quite difficult. So we're also using these much more easy devices, which are needle-free devices, and you can give that either intradermally or intramuscularly. My favorite is intradermal, but we're hedging our bets and checking which of them works better. And then, as I've already mentioned, we are developing a more systemic injection technique, either via peptide or a lipid. So, okay, the idea with this second generation vaccine is by going for N and RBD, that we might get better immune correlates or better immune protection against any variants that are arising. And that's clearly something that we need to think about. Or the alternative is we have already made this RBD variant as well, or we could use that as a separate or a combined immunization. So why would we basically consider making a vaccine when there are already approved vaccines? Well, clearly we started at the same time, but for various reasons, mainly money, we were a lot slower. But clearly, in theory, we're better because we shouldn't have vector immunity. The problem with the RNA vaccines is it's more difficult for them to simultaneously target multiple antigens. They have to have multiple vaccines. So we have an advantage there. Hopefully, by going for two proteins, we've shown you can get both antibodies and T-cell responses to both, which hopefully confers better protection or at least gives some protection against new variants. As I say, we already know in our melanoma patients that we get strong memory responses over long periods of time. It's very stable. And I guess we've gone full circle now because this FC modification that we have developed for this vaccine now works really well in our original cancer vaccines. So our original cancer vaccines are now vastly improved, and we've got new trials starting shortly with those as well. Thank you very much.
Fantastic. Thank you very much. It's a great talk. Very exciting. Just time for one quick question. You know, there's a clear need to vaccinate the world. None of us are safe until everybody is safe. So I'm really fascinated by the stability of the DNA vaccine compared to the RNA. Do you think you'd need refrigeration to transport your vaccine and store it before administration?
Yeah, so we've got, I mean, basically at -20, as I say, we've got 5 years stability. Clearly, if you then do it in the fridge, it's probably more like a year. Clearly, even less than that, if you weeks or months, basically at room temperature, it's incredibly stable. You wouldn't be able to ship it around at room temperature forever, but yeah, much more feasible.
Yeah, much easier than a -70 freezer.
Yeah,
okay.
Definitely.
Fantastic, thank you, Lindy. So I think we'll move on to the next session in order to keep to time. But thanks to both speakers in this session. Great talks, thank you.